Yeah. Thank you for this appeal, and we open the discussion. Yep.
Werner, you ended your talk with the expressed hope that evolution will continue for additional millions of years if we take proper care of our environment to allow that to happen. I know that some people have suggested that human evolution, as opposed to perhaps bacterial evolution, is at an end because of the fact that we have, in a sense, short-circuited the process of natural selection by medical intervention. That is, there's very little early death of human beings because we have such good medical attention. And I'm wondering what your view of that is. Is human evolution likely to continue in the next 3 million years, assuming that our planet stays in good shape for humans to live, or have we really brought human evolution to an end, at least through natural processes?
Okay, well, of course, I had not enough time in my talk to remind us ourselves that this self-organization of evolution requires, of course, a number of genes. And I have started to call these genes evolution genes. And some of these evolution genes are actually variation generators. For example, transposase, which are transposing a segment from one place to another in the genome, and there are many other already known functions. But many of these evolution genes are also inhibiting too efficient mutagenesis. If we would have in all the germ cells steady new mutations made, a majority of mutations are negative, and then that would erase rather rapidly that species. So it's a wonder work which nature fulfills. I see the genes there, they are inactive, and only in one cell of perhaps 1,000, all of a sudden there is a change there. Darwinian selection will see whether that is appropriate or not. If it's appropriate, that's a step ahead of evolution. But I have no doubt that that will function properly. And for me, it's unexpected to see these genes in the cell. They are, for example, I give you just one example. We have taken care, we had isolated a transposable element, which are, many elements are in bacteria, in other living beings, in plants, in animals, in our own body. They can change place within the genome, and these should very rarely do that. Otherwise, it's a disaster. And this element which we have studied, we isolated it from the sequence. It was an absolutely normal expression promoter in front of the gene, and we sequenced that out. We cut it out and put in front of another gene, which we could the product very easily measure, and it was fully active. That was a surprise, because we knew the element jumps very rarely. And then it was also found that in the middle of the reading frame, there is a terminator of the translation, of the transcription translator, terminator. And that term was quite active and terminated almost all, but not quite all, of the transcriptions. The other one, the few which went to the end, gave finally an RNA molecule, and that RNA molecule was inactive because it had, in the other sense, an RNA produced which hybridizes rapidly with the single-stranded transcript and inactivated it by this way. And very rarely, of course, the transposase can do its job. So it's enormous, impressive to see how inventive nature is. All of these things, and I cannot go into more details, I just want to tell you that we have always seen that just in E. coli bacteria, there are, for the same function, not only one, but several systems which all function properly. And it's my expectation that if you extend that research to other living beings, including us, you will see how inventive nature is, and it's not always using precisely the same strategies.
Well, I don't really have any doubt that the processes of genetic variation, whether by mutation or transcription errors or whatever the mechanisms that you've described, that those are still just as active as they ever were. It's the second part which you mentioned, the necessity of selection. Mm-hmm. And the point is that because of so much modern medical intervention, the advantage that some particular genetic variation would have on any individual's ability to reproduce seems to be something that is less of a reality in today's world than it was in the world of our ancestors even thousands of years ago, let alone millions of years ago. And
I agree fully. And I do not expect that in another million of years we have our own progeny there. There are other intelligent living beings and having taken over and perhaps making the same kind of scientific investigations that we have already done. I'm not sure, you know, but it's an open life span, yeah.
Okay, as you mentioned in the introduction of your talk, bacterial diversity within our gut, and the number of bacterial cells are more than 3 times larger than human cells. And we know those bacterial populations are essential for our survival. Also, you mentioned horizontal gene transfer is naturally taking place quite frequently. On the other hand, as we discussed, even in this academic quite number of times, GMO, or genetically modified organisms, I think we are a little bit reaching a different stage of this problem because the scientific advancement is coming much more new discoveries such as human biome and like a natural gene engineering is taking place. Therefore, not only like a dangerous side, but also we have to emphasize on positive side too. On the other hand, synthetic biology is now successfully making an entirely artificial E. coli. So understanding this kind of a situation, what do you think in the future, how we should, as a scientist, behave to the society from your perspective?
Okay, well, of course, there were in the past already a number of conferences. I remind ourselves that in '72 there was, that was shortly before, when genetic engineering had been started to be done, the Asilomar conference, and formulated some kind of rules on what kind of precautions should be done. Nowadays, with the CRISPR-Cas system, there is another debate in the scientific world. This is a way to more intensively, actively edit genetic information on the site. I agree, this is something which has to be dealt with by our academy because it has ethical considerations. And I look forward to a workshop which Raven has proposed to make on extinction. So there we should debate what leads the living world to lose some important living beings, and perhaps we will also consider future possibility for extinctions. So I think it is good that our academy remains to be on the front of present-day research.
You earned your Nobel Prize for the discovery of restriction enzymes. So can you imagine a world before restriction enzymes when there was essentially one superorganism in which DNA was horizontally transmitted between all life?
Yes, actually, Hamilton Smith had started independently to look for endonucleases in bacteria and found some, and then he became aware of our research on restriction modification systems and found that, in fact, what he had already in his laboratory was a restriction enzyme. And that's why we then got the Nobel Prize together, together also with Nathans, who was a colleague of Ham Smith and had applied these available enzymes already to study human cancer virus in his laboratory. Yes, I think we have to be aware that future activities are still going on, and synthetic biology, of course, is also, and people dream. So far, I haven't seen any publication, but many scientists dream of the thing that they wanted to have a protein of for some function which you don't easily find in nature, and which you can then synthetically make. That hasn't brought, as far as I know, any success so far, but sooner or later it may. But I may recall that Eli Biorech, working in Brasilia, had been in contact with me, and he has isolated, okay, we know about 10,000 years ago, the human society started agriculture by domesticating animals, by domesticating a number of food plants. Now, Eli Biorech has introduced the concept of domesticate a particular gene or a set of genes. And the one which was at the early time done with high success was thinking, you know that spins produce fibers, and some of these fibers are very strong, valid, much better than nylon. And you cannot domesticate the spin. That's not possible, and to have enough of that product. But what he succeeded to sort out from this spin a set of genes which were then transferred into a bacterial cell, and by growing bacteria they produce this spin fiber in big amounts. So he calls that domesticated genes. And the idea was that if we are now testifying loss of biodiversity, we should, before all of these important things are lost, sorting them out and putting it in a kind of safe condition where they can serve our civilization for long periods of time. So I think it's good to know all of these possibilities. I don't see any ethical major problem with domesticating these spin fiber genes.
First of all, I want to congratulate you on your tremendous originality in helping to show us so much about the living world. Yours has been a really helpful and constructive career. I would like to call attention to one of the statements that you made that I endorse fully. Namely, one of the reasons that we need to worry about preserving as much as possible of biodiversity, one of the reasons that we need to think as much as possible about preserving biodiversity is we haven't got a clue about the variety of genetic mechanisms present in many of the organisms that are alive now. Even leaving bacteria aside, we've given names to about 2 million of the estimated 12 million species that occur in the world. We're driving them extinct at the rate of tens of thousands per year, and we're likely to lose half of them by the end of this century as population growth, greed, consumption, and so forth change the character of the Earth permanently. But now's the very time to think seriously about the problem that you brought up. Investigate various kinds of genetics. I mean, it's only been 50 years since we even knew the genetic code, 2 generations. It's only been 40 generations since we invented agriculture, and yet we're sitting here at a time when we're losing this enormous treasure that distinguishes the Earth as far as we know from all other planets, and just dropping it down the drain, as it were. It's time to begin to pay attention to that from a spiritual and a general point of view, and it's time to really begin to investigate it in the kinds of imaginative ways that you've helped us to understand so fully.
Well, thank you. When you talked on the genetic code, I just felt I should clarify also that horizontal gene transfer works only because all living beings have the same language. And that's quite interesting. And I'm very curious to know whether on that other exoplanets they speak the same language.
So, thank you very much, Professor Arber.